All right, so welcome everybody. My name is José Soto. I'm a fourth year biochemistry major. My mentor is Dr. Morris Maduro, and my project is studying metabolism in a model organism using an ATP sensor protein. So in our lab, we use the C. elegans as our model system for the genetic studies of biological phenomena, including metabolism. C. elegans are a species of nematodes, and they're these small worm-like creatures. They're about 1 millimeter in size. They go through about 2 generations per week, meaning they have a very rapid life cycle, and they produce lots of identical offspring, making them very useful tools for experiments, in addition to the fact that they're optically transparent, making them useful for when we introduce foreign DNA, such as GFP reporters, to perform our other experiments. So why are we studying metabolism? Well, metabolism is important in daily life. It's a series of biochemical processes that occur within all organisms to sustain life. It's the combination of oxygen and nutrient intake to feed pathways responsible for growth, maintenance, and our ability to function in response to our environment. So this is the process of cellular respiration, where we break down food into glucose, and then ultimately what we turn our food into is this energy that you see here known as ATP, which is produced in the mitochondria. And why study energy metabolism? Well, in the US, we have increasing rates of obesity and diabetes. And these two diseases are very associated with the amount of energy we intake. So why is ATP important, or why is adenosine triphosphate important? So it's the main source of chemical energy transfer in living cells, and it's needed for a variety of processes, including those that you see here, such as muscle contraction, cell growth and division, and cellular respiration. Um, and we're going to try to measure ATP using an ATP sensor that we're going to integrate into the intestine to be expressed into the intestine of the worm. However, the sensor is not a machine. It's made out of fluorescent proteins. Therefore, it is coded by a gene that is not native to the worm genome. Therefore, we must integrate it into the genome so that it can be transmitted evenly across the population. So what we do is we inject that DNA into the worm, and then when we radiate it with gamma radiation, it'll create double-stranded breaks that separate the DNA, and by chance, the gene is integrated. The way the sensor works is through a process known as fluorescence resonance energy transfer, or FRET. So here we have a green fluorescent protein that absorbs blue light and emits green. We have a red fluorescent protein that absorbs green and emits red. And when two fluorescent proteins are close enough together, emission from one will provide the excitation for the other. And this only occurs when they are 10 nanometers apart. So as you can see here in this FRET example, through the initial excitation of the green fluorescent protein, it will then excite the red fluorescent protein, producing that red signal. And this is how our sensor is going to work to detect ATP, because we have a clover and an M-apple protein that flank an ATP binding domain. And activity in this domain will change the sensor's conformation and the distance between the two fluorophores, allowing for that FRET interaction to occur, which I mentioned earlier. So we can excite each fluorescent protein individually, but when ATP is bound to the binding domain, it will allow for that FRET interaction where we excite with blue and ultimately see red. So we need some controls to determine whether our sensor is reliable to detect ATP, because what we know that we're going to get some signal, but we can't actually look at the sensor and know whether or not ATP is being bound to it. So we need to know, is the fluorescent signal due to ATP binding to the ATP binding domain? And to do that, we're going to use a dead sensor that has an inactive ATP binding domain and compare it to the normal sensor. So this dead sensor that you see here has that inactive domain where, regardless of the presence of ATP, will not be able to bind, and therefore not bring the two molecules closer together compared to the normal sensor, as mentioned previously. And we hope that the normal sensor will produce higher signal because it has that activity to bring the two fluorescent proteins closer together. And with this established, we need to determine, well, can the sensor measure changes in ATP? Because if it cannot measure changes in ATP, then how can we tell differences in energy metabolism? So we're going to compare the normal sensor to previously studied genetic backgrounds with known ATP concentrations. So a little bit of insight into what I do in the lab to perform this experiment. We grow the worms on MGM agar plates in small, medium, and large plates, and we need to ensure that none of them are starved or contaminated, and that they're all at the same life stage, which is the young adults, in order to keep our results consistent. For the imaging preparation, I initially bleach a population of C. elegans, so that way we can get a synchronized growth of embryos so that way we have a large population of young adults to image when we want to image, which is approximately 3 days after when the nematodes grow from young adults from the embryonic stage. What we then do is prepare a glass slide where we pipette droplets of buffer. I then move the C. elegans from the first plate onto the first droplet, and then from the first droplet to the second droplet, because the C. elegans carry bacteria on them, and we don't want the bacteria to get inside of the image. And what we then do is secure the slide so that way the worms are not moving, because if they are moving, you'll get a very blurry image, as you can see indicated here. So for the imaging, what we do is we mount that slide onto the microscope, and then we focus and center the worms using a 63 times objective lens. We are then going to take 2 images. The first image, we turn on the fluorescence, and then using a Tritzy filter, we take an initial picture. Then we move the filter over to take another image, and this will be a threat image. So we did all our experiments using these camera settings, and the particular place of the worm that we image is this anterior of the gut that you see here highlighted in orange. So what are we measuring in these 2 images? Well, we want to measure how much of that sensor is there. And to do that, we excite one of the fluorescent proteins, in this case MAPL, with green light and measure the amount of red light, which is independent of ATP binding. In the second measurement, we want to know how much of that sensor is activated due to ATP. And this time, the emission is dependent on ATP. So as you can see, when we excite the cloverfluorescent protein with blue light, only those that have ATP bound will produce that red signal. So here's what the images actually look like under the microscope. On the left, we have the protein image or the sensor image. And on the right, we have the FRET image. And the bottom is just an enhanced version of those you see at the top. So what I then do is I load the image pairs onto Adobe Photoshop, highlight the area of fluorescence, and I look at the histogram for the average red pixel values. I then move those values onto a table to take the ratio of the average amount of FRET over the average amount of Tritzy. And then we then take the average of the ratios of the ratio of the averages. So what this means is that we're trying to take the ratio of the amount of sensor that is being activated due to ATP binding over the amount of sensor present. However, my professor created a Python script which automated the whole image analysis section, putting me out of a job for the image analysis. But it was useful that I did the manual analysis because we needed a control for this Python script to know that what we were producing with the script was reliable. So with this, we can analyze approximately 100 images in a minute, meaning that we can spend more time collecting quality images. So for the first experiment, as I said, we're comparing the normal sensor compared to the dead sensor. And in both experimental cases that we performed, the normal working sensor that had the active functioning ATP binding domain showed significantly higher signal than the dead sensor with the inactive non-functioning ATP binding domain in both cases, which is what we wanted to see. So with this confirmed, we wanted to measure changes in ATP levels. And what we did was we compared to different backgrounds with known ATP concentrations. So ATP3 is a mutation that negatively impacts ATP production, and we saw a decrease in signal. DAF2 overall increases the metabolism of the worm, and it produces higher ATP, and what we saw is an increased amount of signal. And DAF16 produces no known changes to ATP, and what we saw is no change in signal compared to the control. So in conclusion, there were significant differences between the normal and dead ATP protein sensors. The sensor responds appropriately to changes in ATP in different genetic backgrounds. The changes in metabolism in different genetic backgrounds correlates with differences in FRET fluorescence signal. And we can now use this established system, this FRET system, to measure steady state ATP levels in C. elegans in other strains of interest in our lab. And in the future, we plan on continuing to use this sensor with the strains NHR156, which mutate a newly discovered transcription factor that apparently increases the synthesis of fat storage, FASTR1 and FASTR2, which produce novel proteins that may have a role in regulating food intake, and then 3, a mutation that affects early gut development and fat storage. These are all mutations that we believe affect the energy metabolism of the worm, and we suspect that there will be differences in ATP. That concludes my presentation, and I want to acknowledge my faculty mentor, Dr. Morris Maduro, my lab members, the University Honors Program, and the Marquis Stock Program, as well as my funding sources. Thank you for listening to my presentation. And I'm open to questions.
Thank you very much, Jose. That was very